Cells were around for billions of years before the nervous system came along. And the question is whether non-neural organisms have memory. Then I thought, well, what does this mean? It sounds like the synapses aren't where the memory is. The memory is back in the cell body somewhere, and the synapses are simply a reflection of the memory back in the cell body. We do not have a satisfactory neurobiological explanation for any form of memory in any organism.
Good day, you awesome brain-inspired people. I'm Paul. This is Brain Inspired, and you're even more awesome if you support this podcast on Patreon, where you can access full-length episodes, join our Discord community, etc. Check that out if you're feeling generous at braininspired.co. Today, David Glanzman joins me on the podcast. So David runs his lab at UCLA, where he's also a distinguished professor. David used to believe what is currently the mainstream view, that our memories are stored in our synapses, those connections between our neurons. So the idea is, as we learn, the synaptic connections strengthen and weaken until they're just right, and that serves to preserve the memory. So that's been the dominant view in neuroscience for decades, and is the fundamental principle that underlies basically all of deep learning in AI. But because of his own and others' experiments, which David describes in this episode, he has come to the conclusion that memory must be stored not at the synapse, but in the nucleus of neurons, likely by some epigenetic mechanism mediated by RNA molecules. If this sounds familiar, I had Randy Gallistel on the podcast on episode 126 to discuss similar ideas. And David discusses where he and Randy differ in their thoughts. This episode starts out pretty technical, because David describes the series of experiments that led him to change his mind and led him to his current view. But after that, we broaden our discussion to a lot of the surrounding issues regarding whether and if his story about memory is true. And we discuss meta issues like how old, discarded ideas in science often find their way back eventually, what it's like studying non-mainstream topics like David is studying, including challenges trying to get funding for it, and so on. So I link to a bunch of what we talk about in the show notes at braininspired.co/podcast/172. All right, I hope you enjoy our discussion. Here's David. One of the things that I find most interesting about you is that you are a rare specimen in that you have changed your mind about something scientifically. Do you agree with me that that is rare in the world of science? And
to the extent that I've changed my mind, yeah, I think that's pretty rare. And I think it's also, if usually when people change their minds the way I've done and they're older, it's a sign of impending senility.
Oh, we'll figure that out. Yeah.
Yeah. So that gives me pause when I think about that. So
you're not being, that's not serious. You're not being serious, are you?
No,
okay, good. Yes. Well, I mean, I'm getting older,
but
I'm not as sharp.
Let me say that I would perfectly believe that my colleagues would take that view.
Okay.
I would be surprised if some of them didn't take that view.
Well, do you have colleagues that from before you changed your mind that give you a hard time with regard to after you changed your mind?
Not so much. I mean, some people just say, they just kind of roll their eyes. So some of my colleagues. And the people who've known me for the longest time, they don't agree with me, but they take it seriously in the sense because they know me and they know the work I've done. And, I've done pretty good work in the field of synaptic plasticity and learning memory. And so, I think people have to respect that. So at least they know that I'm coming. It's not like I don't know anything about synaptic plasticity. Let's put it that way. I know quite a bit about synaptic plasticity and how that relates to learning and memory in a pretty rigorous way, because I work on a model organism in which you can make very rigorous connections between neural activity, synaptic change, and learning memory. I mean, it is probably the organism that is that gives you the most rigorous ability to make the most rigorous connection. So, I mean, I know something about this, and so I think people who know me very well, they may not agree with me, but I'd say people who take it seriously.
So the organism that you're talking about is one of two organisms that you are studying these days, which is the aplysia,
yes,
the
correct.
sea snail,
yeah,
and which was one of the an organism in which some of the first synaptic plasticity studies were done by Eric Kandel, of course, and others. But okay, so maybe let's back up, and I will have said this in the. Go ahead.
Eric was my postdoctoral mentor. I mean, I started working on aplysia in his lab.
What does he think? What has he thought?
I honestly, I don't talk to Eric much anymore, so I don't know what he thinks. Honestly.
All right. Okay. Well, let's back up. I would have said this I will have said this in the introduction, but we've been talking about you changing your mind, and there's a before and after. And do you want to just summarize? You might be just so sick of summarizing this, but what is it that you changed your mind about?
So we did an experiment where to back up, I've long been fascinated by the work of Karim Nader. And I always thought it would be fantastic to be able to look at memory reconsolidation on a synaptic level and to see later on, if you induced reconsolidation, would the synapses that were strengthened or grew, would they retract? I mean, that seemed to me the logical, that would be the logical outcome of that. And for a long time, I wanted to do that experiment, but I didn't have the people or the resources to do it. And when I say do that experiment, in aplysia, you can take the sensory and motor neurons that mediate the defensive withdrawal reflex, which was the reflex that's been the center of most of the investigations of learning memory in aplysia. You can take those sensory neurons and motor neurons out of the animal, and you can put them into cell culture. They will reform their synaptic connections. And somewhat remarkably, those synaptic connections in the dish have all of the forms of synaptic plasticity, learning-related synaptic plasticity that you see in the animal. So that means that you can do studies of synaptic plasticity in the dish and have confidence that what you're seeing is what's going on in the animal. And there have been a lot of studies that show that that's the case. So the experiment that I wanted to do was induce synaptic memory reconsolidation of synapses in culture, and then look to see if when you block memory reconsolidation by treating the cultures with a protein synthesis inhibitor and reactivating the synapses, would the synapses that grew during the memory be erased? So that's what I wanted to do. And the way you train a synapse in culture is that you apply 5 pulses, 5-minute pulses of serotonin to the sensory motor synapse in culture. The reason for using serotonin is because, due to the work of Kandel and his colleagues over several decades, it's known that serotonin is the neurotransmitter that's released during behavioral sensitization, and that is, it plays a major role in sensitization. So And the reason for using space pulses is because that gives you long-term, and by long-term, I mean 24 hours or more memory. If you put mass pulses of serotonin, you don't get memory that lasts 24 hours. And in this case, the memory is the strengthening, the long-term strengthening of the synapse. And you can measure that with electrodes. You can measure the strength of the synapse. So the experiment we did was we induced long-term, the synaptic analog of long-term sensitization with serotonin. The synaptic analog is called long-term facilitation. So the synapse becomes facilitated, and that facilitation can last 24 hours or more.
It's in the dish.
And then in the dish. And then 24 hours later, we gave a single pulse of serotonin to some of the cell culture dish. And that was the reminder stimulus that reactivated the synaptic memory. That was the idea. And then in some of the synapses that got the reminder pulse of serotonin, we followed that with application of a protein synthesis inhibitor to the cell culture dish. So that's anisomycin. So that is the reconsolidation blockade protocol. And what we reported was that when we tested the synapse 48 hours after the original training, just as Kareem Nader had shown in rats and other people had shown, the synaptic memory was gone. So the synapse had reverted to its original strength. So then we wanted to do the next experiment, which is to actually look at the synapses and see if they disappear when we do the reconsolidation blockade. So we did morphological analogs of this experiment. So we would fill the sensory and motor neuron with fluorescent dyes, and then using, just imaging the neurons in culture, we could count the number of synaptic connections. And the way the stand-in for synaptic connection we used were these presynaptic varicosities that are on the sensory neurons. People who work on mammals call them presynaptic boutons. Mm-hmm. We call them varicosities.
Bulges.
Don't ask me. Bulges. Don't ask me why the difference in terminology. Okay. I never found out. But anyway, so we imaged the culture, counted the number of varicosities, then we gave the 5 pulses of serotonin. 24 hours later, we counted the varicosities, and then again, and then some of the varicosities some of the synapses we gave the reconsolidation blockade protocol. So we gave a pulse of serotonin followed by anisomycin. And then at 48 hours, we recounted the varicosities. And, to my great satisfaction, what we found was that indeed the number of these presynaptic varicosities, which reflect synapses, they strengthened at the 24 hours after the serotonin treatment. And then for the synapses that got the reconsolidation blockade treatment, they went down to their original value. Now, it's important to note that they didn't disappear. You didn't wipe out all the synaptic connections. They just reverted to their original number. So there you have reconsolidation blockade in the dish. So, usually it's a good idea to stop when you're ahead, but we didn't do that. So we said, because we could, we asked the question, well, do the varicosities that get eliminated, are they the same ones that grew in response to the serotonin? Because if memory is contained at the synapse, that's what you'd predict. So we were able to look and follow individual varicosities in cell culture over time. And what we found was that it was totally random. So some of the varicosities that grew in response to the serotonin retracted, but so did some of the original varicosities. And it looked like the synapses were totally uncoded with respect to memory. The only thing that mattered was the number. So at first I didn't believe this result, frankly. And so I made my research associate repeat it, just to make sure it was right. And these were very tedious experiments.
Yeah.
So anyway, she did that. And then I got convinced. So then I thought, well, what does this mean? And I thought, well, it sounds like the synapses aren't where the memory is. The memory is back in the cell body somewhere. And the synapses are simply a reflection of the memory back in the cell body. So we'd also been doing some experiments because I was very interested in the idea that epigenetic changes are involved in memory. And so we did experiments in which we tested the effects of DNA methylation inhibitors, again, on long-term facilitation in the dish, but we also looked at we did corollary behavioral experiments looking at the effect of inhibiting DNA methylation on long-term sensitization. And both sets of studies gave us the same result. If you train an animal and then immediately inject an inhibitor of DNA methylation, the memory goes away and we can't bring it back. One of the things we found was that if you do reconsolidation blockade and test the animal 24 hours later, it looks like the memory's gone, but you can bring the memory back with reduced training that by itself doesn't produce long-term memory, but in trained animals in which you induced reconsolidation blockade, this truncated training gives you long-term memory.
It's kind of a reminder to the organism that this is what you used to do.
Yeah. And so putting all these things together, I thought, well, it looks like what's going on is the nucleus is remembering, and the synapse is a reflection of that memory. It's a functional reflection of the memory. So that was what I started thinking. And then I thought to myself, well, what could be inducing the memory? And it was known that one of the things that is known that non-coding RNA can induce epigenetic change. So I thought, well, maybe what happens when you train the animal, it causes the expression of non-coding RNA. The non-coding RNA produce epigenetic changes, and that's the long-term memory. So I was aware from my background in psychology that there were experiments done in the 1960s showing that you could transfer memory from one animal to another by injecting RNA from a trained animal into a naive animal. Now, those experiments are notorious, and they basically, if you read the introductory psychology textbooks or neuroscience textbooks, they always talk about this as a wrong turn in behavioral neuroscience.
But these are planaria? Is that?
Well, they were done initially in planaria, but then people did similar experiments in goldfish and mice and rats.
Yeah.
And the problem was some people reported success and other people reported failure. And eventually the idea died out and the synaptic idea of memory became predominant. So I decided, okay, well, look, we're going to train an aplesia. We're going to sensitize a whole animal. We're going to extract the RNA out of that animal, and we're going to inject it into a naive animal and see if they sensitize. So I got really excited about this experiment, and I went to my members of my laboratory and told them, let's do this experiment. And they didn't. They were really
Why not?
reluctant.
What was wrong with
Well,
it?
they thought it was a waste of time, frankly.
Huh. Okay.
So anyway, so eventually, though, they came around and they said, okay, we'll give it a try. So they did it, and it worked.
Right. Okay.
So
So
we published.
let me just summarize
Yeah,
like what that was, right? So you trained
yeah.
some aplesia to do a task or to be sensitized. What is it called? A
To be
retract?
sensitive. Yeah,
Uh,
the siphon withdrawal reflex. They have
siphon
this
withdrawal
organ called it.
reflex.
Yeah,
Yeah.
yeah.
Okay. So it's trained, it's sensitized to that, and then you extract its RNA, the trained, trained organism, aplesia,
Correct.
and then you
Correct.
inject that into other naive animals that have not been trained, and all of a sudden they show the same sensitization.
Well, we tested them 24 hours after the injection.
I said all
Yeah.
of a sudden. Yeah.
Yeah.
Sorry.
Right.
24 hours
Right.
later, they show the
Yeah.
same sensitization,
Yes.
whereas
Correct.
when you inject saline or a control basically into other untrained aplesia, they do not show that sensitization.
Yeah. One of the controls we used, physiological solution is one of the controls, but one of our controls was RNA from naive, untrained aplesia. That was the other control.
Right.
Yeah.
Okay. So the take home there is that somehow the RNA that was injected into the untrained naive aplesia induced this sensitization behavior.
Yes.
And then the bigger claim would be that it was a memory of that behavior.
Yes.
Okay, so we'll come to it. Like I mentioned to you beforehand, I'm all confused about what memory means now. Thinking about these things.
Yeah.
But we'll come to that. Okay, but I interrupted you because I just wanted to give sort of a layman's summary of those.
No, that's basically correct. Now, one of the things that we also did is that we did a cellular analog of these experiments. So this was done by a scientist who was a postdoc in my lab, a French scientist named Alexis Bedecaretz. And what he did were two different experiments. One, he took sensory neurons, isolated sensory neurons in cell culture, and treated them with either saline, RNA from trained aplesia, or RNA from untrained aplesia. And what he found is when he tested the animals the next day, they were hyperexcitable, meaning that the amount of current that you needed to inject into the neurons to trigger an action potential was lower. And also, sensory neurons, it's a little technical, but sensory neurons, when you give them a long depolarizing current pulse, aplesia sensory neurons fire at the start of the pulse, but then they remain depolarized, but they don't fire. And in sensitized animals, if you put a depolarizing current pulse, they fire throughout the current pulse. And we found that the sensory neurons that were treated with the trained RNA, but not the control RNA, showed that same effect. When they were, as long as they were depolarized, they fired action potentials. Now, so that was a cellular analog of behavioral sensitization that you see. Jack Byrne and others had shown that you see this in sensitized animals. So then we also looked to see if we would get long-term synaptic facilitation using the trained RNA. And there our results were ambiguous. So what happened was some of the synapses showed large changes and other synapses showed no change. And the mean was no change, but the variance was much larger, significantly larger than we saw in the control animals. So we didn't quite know what to make of that. But basically we weren't able to replicate the synaptic effect of sensitization. But something was going on because, as I said, the variance was much larger in the synapses that got the treated RNA.
So, I believe the effect was specific to sensory neurons and was not in motor neurons, correct?
Correct. Yeah. That's
so
right.
What does that tell you?
Well, in general, you don't see motor neurons increase their excitability with sensitization.
Okay.
So that's similar to what you see in the animal.
So it's more like a quicker pipeline to the response via the
Yeah.
sensory neurons or something.
Yes.
Yeah.
Yes. Yeah.
Okay. So let's see. Where do I start? Where do I start? So kind of zooming out. Then, so you've obtained all this evidence that learning and memory, that the synapse is not where the action is, that it must be intracellular, and to your liking, it's within the nucleus, within epigenetic DNA methylation changes.
It could also be genetic. In other words, there's a type of genetic change called retrotransposition. And we're investigating now whether learning anesthesia involves retrotransposition. So it actually could be. So retrotransposition is when, if you're familiar with it, typically it's when a non-coding RNA is expressed and then it's reverse transcribed by reverse transcriptase and then inserted back into the DNA. Typically, it's inserted into a different place. Often, it's inserted into promoter regions of genes. So this is something that has been documented in the central nervous system. Fred Gage at UCSD, at the Salk Institute, has shown that mouse pups that are maternally deprived, there's an increase in retrotransposition in neurons of the hippocampus. And we think the same thing is happening during learning. And we're looking at that right now. So you could have two types of changes. You could have epigenetic changes, but you could also have genetic changes. And those could encode the memory.
And just to be clear, we're talking about long-term memories here, correct?
Yes, we are. Yes.
Because
not short-term memory.
Yeah, that encoding takes time. And the idea is that you need a more permanent kind of storage for this long-term kind of memory.
Correct.
Okay, so, how, just in a very general, broad sense, how has this changed your worldview about learning and memory? I know that there's the old sort of dogma story about synaptic learning and memory. And in fact, when I was interviewing for graduate school, one of the interviewees just sat me down and said, "So what do you think? Is plasticity important for learning and memory?" And of course, like any good student, I hope so, according to the textbooks, let's first talk about how it's kind of just changed your overarching worldview of learning and memory and just cognition as a whole.
So it's interesting to think about that this was a view of many prominent neuroscientists back in the 60s.
Yeah.
So many prominent neuroscientists, not just McConnell, he's the one who's the best known, but many prominent neuroscientists were very taken with the idea that memory is encoded molecularly as a molecule. And that idea didn't survive, but it's starting to make a comeback.
Well, it didn't survive because of the plasticity stuff in the 70s, which came to the fore, and then arguments, evidence for and against the molecular memory mechanisms, right?
Yeah, but I think there's a larger problem. There's a scientist, a biologist by the name of Günther Sten, who wrote an article on scientific prematurity, and he discussed several examples of ideas that had come along in science but didn't survive. And they didn't survive because the scientific groundwork wasn't there.
Yeah.
And if you think about this molecular encoding idea, many people suspected that it was RNA. But back in the 60s, nobody knew about non-coding RNA.
Yeah.
Nobody knew about retrotransposition. People, to the extent that people thought that RNA might be encoding memory, they thought that memory was encoded by the base pairs of the RNA, just by analogy with genetic encoding in DNA. And that idea is almost certainly wrong.
You say now, but who knows, right?
No, I don't think that idea is correct. But it could be. I mean, I don't know, because I don't know what the actual mechanism is. And so if you compare that with the synaptic idea had all the elements in place. Not only did they have the sufficient knowledge to envision what a synaptic change would be like, but they also had the tools and the technology to record changes in synaptic strength. So if you compare that to the molecular folks, they didn't have RNA-seq. So how could they figure it out? So I think it was partly, scientific. This is an example of scientific prematurity. And now we have the tools, and we have a much richer idea of molecules that could encode memory, and particularly RNA, or at least maybe RNA might not encode it, but it induces the changes that do encode it. And so I think the time is right for a reinvestigation of this idea. And people now are reporting changes in inherited memory in C. elegans and mice.
Yeah.
And in several cases, their RNA is involved in the inherited memory. It's one of the mechanisms for the inherited memory. So the idea is an organism learns something that induces the expression of non-coding RNA that gets into the blood, and that produces epigenetic changes in, for example, the sperm. And so that's been documented. And so I think that's another case where this idea about molecular encoding of memory is coming back. So this is, frankly, you asked how this has changed my worldview. I almost never read articles on synaptic plasticity. I'm totally bored with them. And I find myself reading articles on RNA and the effects of RNA on memory, and retrotransposition and all those things.
Are you bored with those synaptic plasticity articles, or disgusted by them, or they're no use to you?
I just think unlike, I think Randy Gallistel and I disagree on, we agree on many things, but I think one of the things we disagree with is whether or not synaptic plasticity has any role in memory.
Right.
And he just doesn't think that it does.
Yeah.
And to me, it's inconceivable that it doesn't play a role, that synaptic plasticity doesn't play a role in memory. I mean, we can show in aplesia. I mean, it was shown by Eric Kandel back in the 70s that when animals learned, there were synaptic changes. The synaptic changes correlated with the memory, and they were, it's just, there's no way that those are simply phenomenologically non-functional changes.
Mm-hmm.
Where I differ from most of my colleagues is I don't believe that that's the encoding mechanism.
Okay. Okay. So, okay. Well, but then you have to appreciate that there is a role for synaptic plasticity. The curious thing to me is that you're just, uninterested in the synaptic plasticity story now, because at this point, I think you've written about this idea. Well, how are we going to connect plasticity then to these intracellular mechanisms?
Yeah. It's not, look, you have to understand. It's not that I don't think they play a role. And I do think that it's really important to understand if memory is encoded molecularly, as I believe back in the nucleus, then there has to be a mechanism whereby the nucleus is regulating the synaptic strength. There has to be some mechanism. So that I'm very interested in. But first we have to figure out in order to answer that question, we first have to figure out what the encoding is, because we don't know right now. So so that's what I'm focused on. That's why I say synaptic plasticity bores me, because the people who are doing synaptic plasticity studies, they're not interested in this communication between the cell body and the nucleus and the synapse. They're simply interested in the synaptic change as synaptic change.
Yeah. Okay. I was gonna ask you this later, but I'll ask it now. But because when we talk about, excuse me, like a synaptic change, let's say a memory, right, or the encoding of a memory, we think about like one synapse. That's kind of like and then there must be some. So then there's a presynaptic activity. It goes to the postsynapse, and then according to your story, according to the RNA or genetic story, then somehow there's an encoding of that association or that activity associated with that memory into the genome. But in reality, so this is just one slice of the complexity of our brains, right? Yeah. There are tons of different neurons all connecting, right? And of course, we're just in constant motion throughout the day, learning different ways of moving, et cetera. And so presumably then you would have to have, you kind of picture it as like one mechanism. All right, I'll remember that cell over there. I'll remember that axon over there because I need to. Yeah. But really what it would have to be is I'll remember these 10,000 axons, and I'll have to encode all of that in my genome or epigenetically or something. I mean, does that bother you? Is that feasible? Is that?
It definitely bothers me. Is it feasible? I don't think anyone knows. I mean, I think in principle, yes, I think it is feasible. And also the other thing to remember is people say, well, each neuron has 10,000 synapses, so the nucleus is going to have to know 10,000. And really, the reality is probably more like hundreds. And still, that's a lot. Let's say that's still a lot. Yeah. And so I
know that that's not beyond the capacity with the capacity of computation with nucleotides, but
that's right. Yeah. No one's shown that yet. Okay. And I think to me, that's something that would be, really important to show that what I'd like to do, the experiment, but unfortunately right now I don't have the resource to do that experiment, is to repeat our experiment, but just simply show that two different synapses onto the same neuron can be altered by
kind of push and pull.
Yes, correct.
Using those mechanisms.
And then that would make it feasible. I mean, the problem is my colleagues just dismiss this possibility out of hand. Many of them do. And I take it very seriously because I think that the cell really is the cognitive unit. So people talk about talk about neural networks. And I agree there are neural networks, but I actually think the cell is a cognitive unit.
I know plenty of people who would still agree with you, despite the story of modern neuroscience.
Yeah.
Well, okay. So there's the thing is, there's so many different levels, right, of organization, and the more we discover, let's say even about the structure of dendritic trees or something, right? Oh, it's more complex than we thought. The structure of networks. Oh, they're more complex than we thought. And then the structure, the networks within a cell. Oh, it's more complex than we thought. And there are all of these levels of organization. And then, so then I'm just going to immediately jump back to what the hell is a memory, because there's memory in the sense of like a subjective kind of, the experience of recalling something, right?
Yeah.
Let's say.
Yeah.
But then there's the storage of memory. There's the retrieval of memory, the expression of memory.
Yes.
And so often when I'm reading work that is talking about these encoding mechanisms on the intracellular level, and they call it a memory, does it need to be called a memory, or is it's like that bit is part of a memory, perhaps? Or like where do we stop talking about, where do we start talking about a memory? Like at what level?
Yeah. Yeah. That's a very good question. I think the answer to that is really complicated. Let me go, before I get to that, let me go back to this idea of the cell as a cognitive unit.
Okay. You bet.
And
I like this idea, by the way.
Yeah. Okay. So if you think about it, cells were around for billions of years before the nervous system came along. And the question is whether non-neural organisms have memory. And people have, at our meeting, there were people who talked about memory-like phenomena in plants. And Wade Marshall at UCSF is looking at memory in stentor, which is a unicellular organism. He's shown habituation, which is memory. I mean, I think that's another issue we can go into, but it's memory. And so if single-celled organisms, if non-neural organisms can have forms of learning that, everyone agrees are learning, like, sensitization, habituation, and classical conditioning. Well, when the metazoans came along and nervous systems appeared, did they just jettison those ancient mechanisms of memory? That just seems impossible to me. I just don't believe that. That's not the way I think evolution works. And so somehow the synaptic mechanisms of memory became integrated with these non-synaptic mechanisms in some way. And the question is, how? To me, that's the question. How? And if we knew that, then we would be a long way towards answering this question you ask, is how does the nucleus know what's going on at the synapse? Somehow, the nervous system mechanisms of memory, neural mechanisms of memory, if you will, became integrated with the non-neural mechanisms. And I think that's persisted to this day in us. And so if we knew how that happened, then we could answer a lot of these questions, functional questions about memory. Now, the question you ask about what is memory, that is a really difficult question, and you'll get different answers from different people. So some people will only say it's memory if you can show it, if it's associative and you can show it in a mammal. and then other people will say it's like a crease in a piece of paper is memory. It's a persistence of. So other people,
molecules in a rock are memory.
Yeah. Right. So, to me, to answer that question, I guess I would say if it's functionally related to a form of learning, then I would say it's memory.
Well, then you have to ask what learning is also.
Well, okay. So with learning, I'm pretty much of a classicist. So I look at, the standard ideas about learning. So, habituation, sensitization, classical conditioning, Skinnerian conditioning, and then other forms of declarative memory, which, those are quite complicated, obviously.
Well, going back to the idea of a single cell as a unit of cognition, and I leapt and said, "Oh, I really like that idea," which I do.
Yeah.
However, I think that's true, and it's also not true at a different level, right? So, my whole organism, my brain and whole organism, learns to play baseball and can remember how to swing a bat, and my that single cell doesn't know anything about it. Right.
Yes.
So that's a different level of organization and memory, I don't know. I just sometimes when I'm reading the literature, we all do this, use words like learning and intelligence and things like that.
Sure.
So that's a unit of cognition, right? But so am I, right? Some sort of unit of cognitions, and how to reconcile those two things while using the same term, memory.
Well, you have to understand, when you say a single cell doesn't know how to hit a baseball, that's true, but you don't know what role the single cell has in hitting a baseball. And that's because there's a lot that your consciousness is not aware of that's going on.
Yeah.
And so, these days, I have to say that I think the idea that there is a person with a unique personality with coherent behavior called David Glanzman, I think that's a kind of useful fiction that my brain tells my body in order to get coherent behavior. So I really think you're basically a colony of billions of little conscious units.
Yeah.
And somehow that produces coherent behavior. And I would not have said that 10 years ago. So that's another way in which my worldview has changed.
Do you know
somebody after I made this statement at this meeting we just had, pushing the boundaries, and two people came up to me afterwards and said, that's what the Buddhists believe. The Buddhists believe the same thing. And I'm going, I didn't know that, because I never paid any attention to religion.
Yeah, everyone who studies consciousness ends up in Buddhism, apparently.
Yeah, I guess so.
Yeah,
even if you're not aware that you're ending up in Buddhism.
Right. Well, I mean, you said consciousness, which is, let's not go off on that tangent, I guess.
Yeah,
but,
sure. Right.
But do you know who William B. Miller is? He's a cellular molecular, but he has the same view of he really elevates the status of the cell as an individual unit that has lots of intelligence, right? And I think it's really hard to...
I will look at his stuff, because that's exactly that's exactly the viewpoint I'm moving towards. Yes.
I just don't, I think it's difficult to talk about because we're using these single terms and we're really talking about different levels of organization.
Yes.
So when I say, when you say memory, to me, it means lots of, let's say it's encoded in the nucleus, right? The synaptic structures are encoded in the nucleus, the tags with the IDs.
Yes. Yeah.
Well, the memory is not the thing that's encoded. That's like the IDs and like the little things that are required to get the to express the memory. Does that make sense?
Yes, that does. That makes perfect sense. I mean, in a way, I guess. I mean, couldn't you say that about any mechanistic change?
Yes.
Couldn't you say the synaptic change? You could say exactly the same thing about. There's nothing special from the viewpoint you're looking at, because the synaptic change, when people say, well, there are more dendrites, and it's the same thing. And so the memory, then you would have to be an outside observer and say, oh, that's that epiphenomenon, that summary of all those tiny functional mechanistic changes, that's memory, right? I mean, there wouldn't be anything. I mean, I'm trying to think, is there anything that you could look at and say, oh, that's memory, without knowing what the functional product of those changes were?
Yeah, I mean, it's all behavior. You always have to express it in behavior.
I think so.
Well, okay, let's go down this road then. So you've made a conscious decision, or a decision. Your trillions of cells made a decision that
I would say a professionally somewhat suicidal decision.
Well, that's another thing. I was going to ask if your favorite poem is what is it? Robert Frost, two paths diverge. The road less traveled, right? Is that the poem?
Yes.
Yeah.
Yes. Unfortunately, if you want to be a scientist, you have to get funding for your research.
How's that going for you?
That's going not really well. So
why?
So I know. So I told this story at the meeting. So our paper is, do you know altmetric scores of
No,
papers? Do you know
no
what that
PICs.
is?
No, no.
Oh, I'm surprised. Okay, so now these days, altmetric is a way of keeping an index of the social attention that your papers are getting. So it scores tweets, blogs, news articles, and there's some algorithm, and it comes up with a score for you, for your paper.
Yay.
So this is, yeah. So yes, and so you will. I want you to cite my paper because that's going to boost my altmetric score.
All right.
But anyway, the altmetric score for that 2018 memory transfer paper is it varies on each given day, but for the last 5 years, more or less, it's been has the number one altmetric score for any paper published in eNeuro. It's in the top 1% of papers published in the field and at that time. And it's in the top 5% of all scientific papers, period. Okay. Now you'd think that would translate into some kind of funding. One would think.
Okay. Yeah. Well, that's a good point. Yeah.
But it absolutely had the opposite effect on my funding. So when I published in 2018, what my lab I had 2 R01s and an NSF. And I went from that to 0. I went to 0 funding. And so eventually I have to give NSF credit for this. They have given me a couple of grants to look at RNA and memory, and also this memory transfer. And that's to their credit. Because there's no way it's clear to me that I could not get this funded by NIH. The problem is that NSF grants are smaller than NIH grants. So my lab has contracted quite a bit.
Oh, I'm sorry to hear that.
Yeah.
And this is also the kind of work that you actually need a lot of
yes.
person hours, et cetera.
Yeah. So, I mean, I'm totally grateful to NSF. It's just that I couldn't keep the lab that I have going with NSF funding alone.
So you're going to try to bring your that metric down, that social media metric? I think you should try to bring it down. You try to get canceled in social media, and that'll. They can't be related though, right? But there must be like
no, it's not related. No. There are plenty of people whose papers have high altmetric scores that are well funded. Sure. It's just kind of, I just consider it somewhat ironic that a lot of people consider this paper to be important, and NIH just doesn't consider it to be important at all. So, no, not important enough to put money into it.
What are your current thoughts? So I was going to ask you this later. I'm going to ask you now. Like then, since we're on the topic.
But I want to say something about this because there's. I don't know if you're familiar with this, but there was a paper that came out in Nature in January, and it plotted the ratio of so-called disruptive scientific publications.
Okay.
These are publications that eventually changed the field. And they use an algorithm to decide whether a paper is disruptive. And typically it's disruptive if it starts out with a very low citation rate and then gradually gets stronger and stronger.
Yeah.
Okay. The ratio of that over the last 50 years has declined dramatically.
The ratio of that to non-disruptive.
Yes.
But is it normalized by just the sheer volume of publications as well?
Well, that's the point.
Okay.
So it turns out the number of disruptive papers has more or less remained, the absolute number has remained constant in a field, but the ratio as a percentage of the publication has declined dramatically. Also, the number of new patents has declined dramatically in various fields.
One way to interpret that is that there's too much money being put into science.
Yeah, that's one way to interpret it. the other way to interpret it is say, well, look, there's a fixed number of creative people in the world, and those drive the field. And there's a whole bunch of other people who are doing stuff that's just really unnecessary to the field. And the fields can remain healthy as long as you have this fixed number of creative people. So some people have said, well, there's no problem here, right? So, there's always going to be new ideas coming along. But a lot of people think it's potential. It really is a problem. And part of this, and of course, people are trying to figure out what's the reason for this. But to me, it's almost certainly the case that some of it is due to the funding mechanisms that are available for science. You can't do science without money. And the peer review system is a fantastic system. It's resulted in America becoming the greatest scientific enterprise in the world, but it's not very good at supporting radically new ideas. In a sense, you can think, well, if you have a radically new idea in a field, you don't have any peers anymore. Yeah. You're outside your peers. And so peer review is going to fail at that point.
Are you worried that you're going to become a starving artist?
I started out as a starving
That's what I am right now.
And I may end up as a starving. When I was a young man, I lived in literally a tenement apartment on the Lower East Side in New York. And I was working as a messenger boy for a film company. I paid $66 a month rent for my apartment. Yeah. And it probably wasn't even worth $66 a month.
Well, I don't know. Maybe the rats thought so.
Yeah. Well, the cockroaches did, and I even had bed bugs. So it's pretty.
But I know. But seriously, I mean, does this worry you about continuing a lab moving forward, or?
Well, continuing a lab definitely, but you also have to understand that I'm in a kind of privileged position. I'm a full professor at a major research university, and I have tenure, and so at least I'll be able to, go to work every day. And so, but yes, it bothers me a lot. The frank answer to that is yes, it bothers me a lot.
I mean, there's the other issue of just momentum in science, because if you don't get funding, right, things kind of come to they slow down, and then you kind of lose the thread of what you're doing, and it's hard to restart, right?
Well, it's not only that. I mean, one of the metrics that people use when they're deciding whether or not to fund you is your rate of publication.
Sure. Yeah. Yeah. You got to start writing a bunch of opinion pieces. Yeah. Yeah. Well, I'm sorry to hear that the funding's not going that. Do you have advice then for, I've often had people, I remember like Paul Chizek saying, because he's a cognitive science person, neuroscience person, and he's kind of going off on this tangent in his career about how following the path, reverse engineering evolutionary stages to help us understand cognition and what brain areas we're doing and how they led to what we're doing now, et cetera. Yeah. And he's super interested in it, but he gave a word of warning, to like, well, you can't go too far off the beaten path because you need feedback from your cohorts, and then you might not make it back, right? But in your case, this is the path. This is and you feel confident about it.
Yeah, I feel confident. And the other thing is that there are people out there who are moving in the same direction.
Yeah.
So, Sam Gershman, Randy Gallistel, he's doesn't have a lab, as far as I know, but he's still very active. And Colleen Murphy is working on memory transfer, inherited memory, but she also showed lateral transfer of memory in C. elegans. That she showed by RNA. Mm-hmm. So there are people moving in this direction. And so that excites me. So I don't feel like I'm this person who's shouting in the wilderness. It's just that I have new colleagues now. So that's, I don't, I just have different colleagues.
Yeah.
And it's not as big a number, obviously, because there aren't a lot of people in this moving in this direction. But the idea that there's learning in single cells, Wade Marshall's work, and the people who are looking at learning in Audrey Dussuteur, who's French, who works on learning in slime mold, she's shown really quite clearly that slime molds habituate.
Yeah.
So, there are people who are in this area, and to me, that's really exciting. So what I'm trying to do is promote that area as much as possible.
It'll come around again, right?
It always does.
Yeah.
It always does.
A moment ago, you mentioned how when you're off on your own doing something, or when it's a radical new idea, right? But this is not. And then before that, you mentioned that there's historical precedent for this,
yes,
and that it got replaced, quote unquote, by the synaptic plasticity hypothesis.
Correct. Yes.
Why? And you mentioned that in textbooks, they maybe mention that and say, well, this was the wrong idea, this was the wrong path. And I don't even remember this when I was learning about learning in memory, and maybe there was something like that, but there are so many ideas that seem to come back when, like you said, the technology is available for actually studying it and in a reasonable and tractable manner. Why aren't ideas like that presented as alternatives in the beginning to students who are, because we're just taught dogma most frequently, right?
Yeah.
So why isn't it given some sort of, at least in the Parthenon of ideas, and given some historical context that the story has not always been just synaptic plasticity, right?
Yeah, I think that's a difficult question to answer. There's a saying I heard once, that the problem with having too open a mind is that your brains might fall out.
Good.
I think scientists are very keyed in to figuring out the truth. And so in order to do that, you have to discard a lot of ideas that you think are non-truth. And I don't think you can have too open a mind when you're trying to push a field forward.
Mm-hmm.
Scientists don't generally say, oh, I think it's this, but it could be this, or it could be that, or it could be that. That's not the way scientific papers are written.
Right.
And to a large extent, that's not the way textbooks are written. Although there, you have a certain amount of freedom to speculate.
Yeah. Depending. Yeah.
I think that's I think that's the, do you know Ignaz Semmelweis? You know who he was? Have you ever heard of him?
You jog my memory. Maybe not. Yeah.
Ignaz Semmelweis was an obstetrician at the major hospital in Vienna during the 1850s. And at that time, women died a lot of childbirth, of fever infection during childbirth. And somehow Semmelweis came up with the idea that this was caused by putrefaction that was under the fingernails of surgeons, because they would dissect corpses and then they would go up and deliver babies.
I do know who this is. I didn't know the name. I know the story.
Yeah. So, and he noticed that his fingers smelled badly when he finished dissecting a corpse. So he experimented with solutions that would get rid of that smell. And what he came up with was a weak solution of carbolic acid. So thereafter, whenever he did dissections, before he delivered babies, he would wash his hands.
Yeah.
And he insisted that all the other obstetricians on his ward do the same thing. And the rate of death of women on his maternity ward decreased to almost zero. So Semmelweis presented his findings to the Viennese Medical Society, and they were outraged,
right?
Because he was claiming that they were killing their patient.
Yeah.
So they basically didn't renew his contract. He had to go back to his hometown of Budapest, and he continued to document the effect of simply washing your hands. Now, the problem was this was before the germ theory.
Yeah.
So nobody had a mechanism. All he had was the data. So in order to convince people, he published a monograph, and it was semi-hysterical, and he basically accused all the doctors in Europe of being murdered.
Well, there's a political aspect to this.
Yeah. So this didn't make him popular. And his wife, who was a socialite, was very unhappy with him. And she thought he was exhibiting, unstable behavior, which he probably was. And so she tricked him into going to a psychiatric institute. Oh. And when he realized that he was in a psychiatric institute, he tried to get out, and he was beaten by the warders, and he died. He was effectively beaten to death. And then, of course, 20 years later, Lister, Pasteur came along, and they came up with a mechanism for what he had found. But there's no question that that he was right, absolutely right. So, whenever I think about my situation, I always think, well, it could be worse. It could be Semmelweis.
I know you could be. The headline would be Dr. Glanzman dies trying to escape the asylum after claiming all synaptic plasticity hypothesis advocates are murderers.
Thought murderers.
Thought murderers. Oh, that's good. Oh, yeah. I hope it doesn't come to that, of course. Yeah. Let's I still have so much to ask you. I want to get back. Let's get back a little bit to the science of it all, and thinking about the scale, right? So you on purpose you chose simple organisms to study, and this has been the dominant organism type, simple, quote unquote, with brains or nervous systems that are comprised of less than billions of neurons, for instance.
Yes.
Well, thinking about the scale of what this would apply to, what it would imply with regards to higher cognitive organisms, right? Like if I have a buddy who loves Tolstoy, I love Dostoevsky, we slit our arms and became blood brothers, and I still don't know Tolstoy, right? So what am I doing wrong? Is the question. But seriously, so it's these behaviors in these organisms that get transferred, which we're calling memory, are fairly sensitive, or fairly simple behaviors, right? Sensitization, reflexes, etc. Where do you see this scaling up? What is your vision for how?
Okay. Yeah. So people have asked me that, and they say, okay, look, I'm willing to buy that. Maybe habituation, sensitization, classical conditioning, and operant conditioning can be encoded molecularly. Mm-hmm. I'm willing to buy that. But I'm not willing to buy that, my memory of Shakespeare's plays, or my memory of the gift I got for my 10th birthday is encoded as a molecule. I'm just not willing to buy that. And I think that's fair enough, because I don't, I personally cannot imagine how that kind of memory could be encoded molecularly. Having said that, I don't see how it can be ruled out. I mean, just because I can't imagine it doesn't mean that you can rule it out. If the capacity, if molecules can indeed encode, the complex information, then I don't think you can rule it out. Now, I personally don't spend a lot of time thinking about complex cognition. Mm-hmm. I mean, I've always been a reductionist in my approach. So I'm not a reductionist in terms of the application of my knowledge. So, what I learn about simple organisms. So I'm very focused on trying to figure out memory in so-called simple organisms. But it's important, and this is something that people, I don't think in the field sufficiently grasp, is we do not have a satisfactory neurobiological explanation for any form of memory in any organism. Now, think about that. Think about that. Okay. We cannot say how C. elegans habituates. I mean, we know a lot about it, but there's a lot we don't.
Famously, we know its entire connectome.
Yes. And that's why this connectome enterprise, which is getting tons of money and attention.
Murderers. Just kidding.
No. It's not to say that it's not important. I mean, one of the major advantages of aplesia was we had a primitive sort of connectome of the nervous system. We didn't know everything, but we knew enough. It's important, but I think many of the people working on that think that the solutions to cognition and memory are going to fall out of knowledge of the connectome. That's certainly incorrect. That's certainly incorrect. I kind of, this will get me in a lot of trouble, but I sometimes think the people who work on connectomes are really smart people who do that in lieu of coming up with really creative, profound ideas about how nervous systems work. Now that's going to get me in.
I'll leave that alone.
That's going to get me in a lot of trouble. But so if you realize that, then for me, it seems that the driving ambition should be to try to figure out how a form of memory works in some organism. And once you know that, then you can try to build your way up. So that's the way in which I'm a reductionist. It's not to say that I think that the mechanism that we find for habituation in C. elegans is going to be, important for all forms of cognition and, declarative memory in humans. I don't believe that at all. It's just, if you take what I'm saying seriously, then you realize how little we truly understand about the brain.
Yeah, it's a depressing thing.
If we can't figure out how we can figure it out, I'm convinced, but if we don't know how learning works in C. elegans, an animal that has 325 neurons in its central nervous system, and we don't know that, how long is it going to take before we understand cognition in humans? Just how long? Just try to figure it out.
Yeah. I'm preparing a job talk for tomorrow, actually, and it's the same old story, right? You think, oh gosh, we know nothing about the brain still and cognition, but then you look back at I'm preparing this talk and having to go through and prepare figures of what I've done, and I've done so much, and I still. And you learn so much, and then mostly what you learn is how much we don't know. And I mean, it's just this catch-22. It keeps going.
When I was in my 20s, I was convinced that synaptic plasticity explained learning. And I thought, okay, look, this is not going to be hard. I'm just going to figure out everything there is to know about synaptic plasticity, and then I'm going to understand learning. And I thought I would finish that certainly by the time I was 50.
Yeah. But what was it? Was it Nader's work? What was it that sparked the change? The beginning of the
thinking about Kareem Nader's work really was the thing that it didn't. Kareem Nader's work didn't make me think that synaptic plasticity wasn't the answer, but it drove me down the path to doing the experiments that led me to where I am now.
Okay. Yeah. Offline, before we started talking, I asked if you had any thoughts about artificial intelligence, because the memory mechanisms that you're interested in are far, far different than neural networks.
And I think that deep learning it's not a bad way to teach machines, and obviously they will learn, and they will learn pretty interesting things, but that's not the way the brain learns. Okay, I'm convinced of that, and that's not the way the brain encodes memory. I mean, I always say, if you could show me a computer that's as cognitively sophisticated as my snail, I'll be impressed. And so far, there is no computer that's as sophisticated cognitively as my snail. In other words, if you put a deep learning computer out in the ocean and it had, let's say you kept it from rusting and it had to deal with all the things a snail deals with, a marine, it couldn't survive. It could not survive. So until we understand the real mechanisms of encoding memory, we're not, everybody gets all excited about, chat GPT and generative AI. And I just say, look, this is not the way humans learn. This is not the way they remember. If this is not the way they remember, it's unlikely it's going to replace us anytime soon.
Yeah, well, there's the replacing aspect, right? Which it's just silly to me, but it is interesting to see how many people are worried about it. But then there's what is it? It is impressive as a tool, and
yes.
So what does that say? What does that say about our cognitive, or what does it say about what we deem to be cognitively awesome, like chess and like language? I mean, does that reconfigure how we should be thinking about intelligence and cognition?
I honestly don't think so, because I don't think that that's the way we do it. I mean, I would, rather than, look, there's a functional value to generative AI that's undeniable. I mean, it's going to be able to increase our capacities enormously. But if you really want to figure out how humans think and remember, this is not how they think and remember. This is just not how, in my opinion, this is not how they do it. It's a very artificial system, and the kinds of things it can do are increasingly sophisticated, but they're still fairly artificial, in my opinion. You probably are too young, but in the 80s, I think, or maybe 90s, when Deep Blue beat,
I think it was 97, right? Maybe.
Yeah. Okay. Yeah. So beat Gary Kasparov in chess. Everybody said, okay, well, now computers are smarter than people. Yeah. Okay. So then I think it was Nike came out with an ad a series of ads where they had Deep Blue in a gym playing against David Robinson.
Oh, nice.
And David Robinson was dunking over Deep Blue, and he was stealing the ball. Deep Blue is just a box. For
you kids out there, David Robinson was one of the best basketball players in that era, in the Deep Blue era.
Yes.
Professional basketball
in the Deep Blue.
The Admiral.
So then they interviewed David Robinson, after he played deep. What do you think? And he goes, well, I think he's got to work on his ball control. And,
Yeah, that's good.
So that's what I think of when I think of generative AI. Yeah, it can do things, and increasingly impressive things that are not so rigid. The formal demands are not as rigid as chess. They're much more flexible. And so it's getting more impressive. But, I just simply don't believe that that's how organisms learn.
Well, all right.
Remember.
So you turn one of those off and everything stays unchanged, right? The weights stay unchanged and everything's stored, quote unquote, in the synapses. You turn it on a year later and it performs the exact same. Is it just a matter of adding some sort of nucleus to the units, some sort of internal storage mechanism that tags the connection strengths and and then you can kind of sever things and it'd be okay.
Yeah, I guess that is true. But then I think you have to get to the bigger question, the one that you alluded to, is how do you get all these independent units to cooperate? How do they cooperate? How do they know? All right. Now, you might think, oh, well, that's easy. They're connected to each other. So it's synapse. But it turns out that neurons have ways of communicating with each other non-synaptically. So I don't know if you're aware of the recent findings of Jason Shepard and Vivian Budnick, but they looked at a well-known synaptic protein called ARC. And ARC has been known to be involved in synaptic plasticity for 20 years, and it regulates AMPA receptor trafficking, and it maintains synaptic strengthening. That's one of the things it does. Well, what Jason and Vivian Budnick found is that the ARC protein is a capsid, just like HIV,
like a virus,
and it contains like a virus, and it contains RNA, and it's shuttled between neurons non-synaptically.
Is it at the synapse, or is it actually extrasynaptically? It's from non-synaptic sites?
It's that I don't know. It is at the synapse.
Okay.
But I'm pretty sure it can be released at non-synaptic sites.
It seems like a efficient transfer would be at the synapse. So
yeah, but it's not. It's taken up by the receiving cell.
Yeah.
By endocytosis. So
it doesn't have anything to do with the action potential, is what you're saying.
Yeah.
Right. Yeah.
No, it does have something. The release does have something to do with action potentials. Whether or not it can be released without action potentials, I don't know if that's clear.
Yeah, I meant like the synaptic strengthening and weakening at the pre and postsynaptic mechanisms, right, that are purported to strengthen and weaken. Is that what you're? Yeah. It's more like just here's some information. Go, and the idea is like.
Yes.
It's just a messenger kind of.
Yeah. Okay. That's true, but it's RNA.
Yeah. Right.
Think about that. So it's RNA. And what if that RNA gets transported back to the nucleus? And how many of these proteins does the nervous system have? And if ARC is released extracellularly, then that means, in principle, it can go out into the extracellular medium and into the blood. So now you have activity-dependent release of RNA that's getting outside the neuron itself and in principle, outside the brain. So what's the function of that? What's the cognitive function of that?
Open question.
Okay. Yeah. So, but going back to single cells, if single cells existed at one point, why wouldn't RNA be a mechanism of communication among single cells? And why couldn't that be used to coordinate their behavior? And the answer is there's no reason why it couldn't, and it probably is, and it may well be the primitive. Before there were neural networks, there may be functional networks of individual cells whose behavior is coordinated by release of RNA and probably other molecules as well.
Yeah. All right, sticking with the single cell for a moment. So the classic textbook story is DNA encodes life, right? DNA encodes RNA, encodes proteins, and that's the central dogma, which we know is there are lots of different ways that can go. But my question is, so still the central story in evolution as well is that DNA like controls who we are and controls the cell, right? But I think a really nice, I love these inversions, these thought experiments that are inversions. One could say that it could inverse that and say it's the cell that's actually controlling the cell as a cognitive unit, let's say, that's using the DNA, that's controlling the DNA. Is that a healthy way to look at it from your vantage point?
So, this goes back to evolution. So, some of a couple of speakers at this Pushing the Boundaries meeting talked about the interaction of the environment on DNA and how the changes in DNA weren't really random. They were guided by some of these mechanisms.
By environmental mechanisms?
Yes, by environmental mechanisms. And so that suggests that it's actually more interactive. So the DNA isn't this master switch. The DNA is acted upon by other factors.
Yeah.
And so I think that's a more realistic view of
Kind of a
things.
circular causality
Yeah,
viewpoint.
right. Exactly. Yes. Yes.
David, in the last few minutes, I just have a couple extra questions for you if you have time for them.
Sure.
We've talked about your struggles walking this lonely, solitary.
It's not so lonely or solitary.
Well, okay, all right.
Yeah, I mean, it's not exactly.
Okay, instead of 10,000 friends, you have 50 in the something like that, right?
That's right. That's right. That's right.
What are some of the benefits, though? Do you feel? Oh, I don't want to put words in your mouth. So, like, what is it that you gain from doing that?
The benefit is I'm meeting a whole new group of people, and I'm very excited about their ideas. And this, this keeps my juices flowing.
Is it fun to go against the grain also, or is that part of the fun?
For me, this is something I have to discuss with my therapist. But it's always been difficult for me to take orders from people.
Yeah,
and
me too.
So there's probably something in me that is, there's some people who they just want to know where everybody else is going, and they want to do the same thing, but just maybe be at the front of the line, but they want to keep doing the same thing.
I don't like lines.
I've never been that kind of person. So there probably is something I'm almost certain there's something about my personality that would mean that I'm the kind of person who would go this way. And also, it takes a certain amount of self-confidence, which, I have that as well. So that's, but, I just, let's set aside the funding issue, because for me, that is a serious issue, because, I'm a laboratory scientist, so I'd like to have a lab. But I just find I'm really grateful that I am going in this direction, because for me, it's endlessly exciting, and I'm learning stuff that I didn't know. I mean, I this is kind of embarrassing, but I never took molecular biology in college because, I was a psychology major, and I thought, what are molecules going to tell you about thought? I'm going to study psychology, and so everything comes back to bite you in the end. But, I'm learning all this really interesting stuff. And for me, it's really satisfying.
Speaking of grants, when yours gets funded, what's your vision for the future? What are the next steps for you once you can go into overdrive?
Well, I think it's pretty obvious. I mean, what I'm going to try to do is figure out the role of RNA in learning memory. And I want to figure out, is it, is memory encoded epigenetically? Is it encoded also partly genetically? What role does RNA play in nuclear encoding of memory? And how does the changes in the nucleus, how does that structure the synaptic changes and keep them maintained? And for me, that's what I'm interested in.
Or reconstitute them, right, when they're not maintained.
Or reconstitute them when they get destroyed. I mean, we showed that when we had reconsolidation blockade and we eliminated the memory, supposedly, we were able to bring it back with this truncated training. Well, the synapses came back too. So that means that the synaptic structure can be reconstituted. And by the way, that would give me hope if I were working on Alzheimer's, because, in principle, if you understood that process, you could reconstruct the synapses that are destroyed during Alzheimer's, and retrieve the memory, get the memory back. Now, of course, when the cell dies, that's it. When the neurons die, that's the end of the story. But certainly for years, the major problem people have with memory is that there's synaptic loss. But if you could reconstruct the appropriate synapses, you should be able to get the memory back. So the memory should still be there. So for me, that's part of the future.
All right, David, I'm sorry to hear that you're going senile. but I've really enjoyed my time here with you. I'm joking. Sorry, I had to throw it in there.
No. That's totally fine.
No, I really appreciate it. It's a really fun conversation, and I wish you the best in funding. And of course, keep up the good work.
Thank you. Thank you very much. And I've enjoyed this conversation immensely as well. Thank you for your questions. A really thoughtful, very thoughtful question.
I alone produce Brain Inspired. If you value this podcast, consider supporting it through Patreon to access full versions of all the episodes and to join our Discord community. Or if you want to learn more about the intersection of neuroscience and AI, consider signing up for my online course, Neuro AI, The Quest to Explain Intelligence. Go to braininspired.co to learn more. To get in touch with me, email paul@braininspired.co. You're hearing music by The New Year. Find them at thenewyear.net. Thank you for your support. See you next time.